I'd like to thank organizers for giving me this opportunity to introduce our GWAS study of gastric cancer and duodenal ulcer-derived H. pylori strains, which was published in Microbial Genomics last year. GWAS, or Genome-wide Association Study, compares case and control genomes and has the advantage of being able to reveal genetic variations associated with a particular phenotype in the absence of any hypothesis of candidate genes. GWAS in bacteria has been difficult due to the strong population structure, but methodological developments in the last several years have helped to control this effect and to systematically explore relationships between a phenotypic trait and any genetic variation. Today, I'm going to talk about GWAS on H. pylori. As you know, H. pylori is phylogeographically differentiated and highly recombining toward the genome by mutual homologous recombination in the stomach. We reasoned that this highly recombinogenic feature would make it an ideal subject for GWAS because the high repeated recombination decreases linkage disequilibrium or correlation between SNPs so that the responsible SNP would be separated from the linked SNPs that otherwise would appear as false positives. And previously, GWAS on H. pylori compared genome sequences of strains isolated from patients with gastric cancer and gastritis in Europe. The phylogeographically differentiated distinctive population structure of H. pylori requires GWAS to be conducted separately for each population. In this study, we focus on East Asia population because it has been associated with the highest instance of gastric cancer. The highest instance of gastric cancer is often explained by the East Asian type CAG-A protein. However, it remained largely unknown whether there are other virulence factors in addition to CAG-A. East Asia is suitable to explore this question because frequency of strains carrying the CAG genes is nearly 100%, compared to 60% in other regions. On the other hand, the previous study in Europe found gastric cancer was typically associated with the CAG genes as expected. In addition, the transition from gastritis to cancer is not discrete but continuous. And the previous study did not elucidate how amino acid changes in the GWAS hints underlie the pathophysiological alterations. In this study, we focused on duodenal ulcer rather than gastritis as a reference for comparison with gastric cancer. Duodenal ulcer, or DU, is also caused by H. pylori, but is considered divergent and even somewhat mutually exclusive from gastric cancer, or ZC. DU and ZC are known to arise from different gastritis conditions. And cohort studies have revealed that patients with a history of DU have a considerably reduced risk of developing gastric cancer. However, no genome-wide study has explored a genetic marker for the discrimination of DU and ZC. The discrimination at an early stage is important because the treatment guideline is different. In this study, we explore what are the potentially multiple underlying genotypic factors in H. pylori that determine the risk of DU versus ZC in infected patients and would allow for discriminating between them. Here you see a core genome phylogeny of 240 strains from HSP East Asia population, mainly Japan and Vietnam. Among them, 209 strains were newly sequenced in this study, and 125 strains were isolated from GC patients. The tree comprised two large clusters of mostly Japanese and Vietnamese strains, and showed no cluster was solely associated with GC colored in red or DU colored in blue. There was no statistically significant difference in disease status frequency between Japanese and Vietnamese clusters. Using the dataset of these 240 strains, including a complete genome of a Japanese reference strain isolated from this patient, we first conducted SNP GWAS targeting approximately 150,000 SNPs with minor allele frequency higher than 5% after adjustment for population structure. This QQ plot shows the p-values of most SNPs were as expected under the null hypothesis of no association. This indicates the absence of systematic inflation of p-values after adjustment for population structure, or genetic relatedness among strains in HSP East Asia population. At the same time, we found 23 outlier SNPs with p-values less than 10 to the minus 4 deviating from the null hypothesis. These p-values were not very small, but to test significance of the outlier SNPs, we conducted the permutation test and found on average 12 SNPs were likely false positives, showing these p-values under the randomly permuted phenotypes. We thus excluded 12 less significant SNPs from the 23 SNPs and used the remaining 11 SNPs for further analysis. The 11 SNPs showed from 12 to 29 frequency increase in the GC population. We also conducted a SNP GWAS using another program, PyTher, which can adjust for population structure. It yielded p-values that were highly correlated with those in the previous slide. Again, most p-values were as expected under the null hypothesis of no association, whereas there were more outlier SNPs deviating from the null hypothesis, including all the above 11 SNPs colored in pink. In addition, we conducted GWAS targeting approximately 1.3 million uniques or unique DNA substrings of variable length, and the QQ plot seems to deviate from the null hypothesis much more than that of the SNP GWAS, as seen in the previous slide, possibly because of more potentially highly correlated features in the unities that may not be capturing signal in the SNPs. Therefore, the p-values might not be directly comparable with each other. In the QQ plot, we focused on outlier unities with p-values less than 10 to the minus 5. And after the permutation test, we identified 14 unities that were grouped into 3 motifs located in 3 different genes. Here you see location of the 11 SNPs and 3 DNA motifs associated with GC on the sequence of the Japanese reference strain isolated from a GC patient. And clearly, there was no peak, including multiple feeds, under linkage disequilibrium. Here you see genes carrying the 11 SNPs and 3 DNA motifs, and some of which I will explain in detail later. Regarding the DNA motifs, each DNA motif included multiple SNPs, as shown in this nucleotide sequence alignment of one of the 3 genes, reflecting the high genomic diversity of H. pylori. In this example, the presence or absence of DNA motif approximately corresponds to C or T at the end of this motif, although the correspondence is not complete. Regarding the other 2 DNA motifs found in unique GWAS, the correspondence between their presence or absence and specific amino acid changes was not clear, indicating difficulty in applying unique GWAS to H. pylori. Overall, the top-ranked SNPs in SNP GWAS were not detected in unique GWAS, suggesting SNP GWAS should mainly be used in H. pylori. For discrimination between DU and GC using the set of SNPs and DNA motifs, we used a random forest model to predict the probability of strain being isolated from a GC patient. using presence or absence of each SNP or DNA motif times minus log 10 p-values of the SNP or motif as explanatory variables. We then conducted a twofold cross-validation in which the data was split into training and test datasets. And the random forest model was fit to a training dataset, and the probability of a strain being isolated from a GC patient was predicted for each strain in a test dataset. Training and test datasets were prepared in 3 ways to check the robustness of results. Performance of the discrimination is shown here as ROC curve, in which x-axis is specificity and y-axis is sensitivity. An area under the curve, or AUC, indicates a discriminatory capacity. Under the optimal cutoff, AUC was 0.88, and the false positive rate and false negative rate was 0.2, respectively. This result was obtained when the first half of strains in the phylogenetic tree was used to train the model, and second half for prediction. We obtained similar results by 2 other means of cross-validation, in which Japanese strains were used to train the model, or Vietnamese strains were used to train the model. Finally, I'm going to explain how discriminatory amino acid changes in the genes suggest molecular mechanisms underlying the disease phenotype. We placed the amino acid changes on predicted protein 3D structures to analyze their function and biological significance. The best example was TLBC, a chemotaxis receptor which binds to lactate. Lactate promotes H. pylori growth in the stomach and was recently identified as a highly used molecule in the stomach. Amino acid change corresponding to the discriminatory SNP causes electric charge alteration at the ligand binding pocket, which may affect its ligand binding properties and may in turn influence H. pylori growth or survival in the stomach. Furthermore, we identified 2 new virulence factor and oncoprotein candidates. The first candidate is ISP, which is structurally similar to the HTRA family of serine proteases, but lacks active sites. It potentially interferes with human HTRAs, maintaining protein quality in mitochondria. And disturbances in their action, are known to be associated with oncogenesis. The discriminatory amino acid change causes electric charge alteration at the protein binding area, presumably affecting protein binding. The second candidate is TRIH, meaning triple halves, which carries 3 half pathogenicity-related domains. The domain may interfere with apoptosis and induce oxygen radical production and affect cohesion in human cell. In summary, we conducted a small sample GBATs using 125 GC cases and 115 DU controls, followed by prediction of GC and DU. We identified 11 SNPs and 3DNA motifs that combined allowed effective disease discrimination. They were informative of the underlying molecular mechanisms. And we also identified novel virulence factors and oncoprotein candidates. These results provide both defined targets for further informatic and experimental analysis to gain insights into gastric cancer pathogenesis and the basis for identifying biomarkers for distinguishing these H. pylori-related diseases. I would like to thank my co-authors and reviewers and these collaborators and a national grant to support genome sequencing and supercomputers in National Institute of Genetics in Japan. Thank you very much for your attention.